Hello, everyone. My name is Stephen Dobson. I'm a professor at the University of Kentucky, as well as president of a spinoff company named MosquitoesCOST in Lexington, Kentucky. And I'd like to start by saying thank you to the conference organizers for the invitation and the opportunity to present. I very much wish that I could be there in person, but things as they are, I was unable to. So I appreciate being allowed to make this virtual presentation. So I'd like to start by acknowledging all the different groups that we've been working with. There's just not enough time to give adequate discussion of each of these participants. But again, I'm with both the University of Kentucky and MosquitoesCOST. We've been collaborating with a number of abatement districts here in the United States. I'll be speaking about some of them in a few moments. Of course, funding that we received from the CDC and from the National Institutes of Health, as well as the Gates Foundation, and then collaborators as well that I'll mention in just a few moments. So I have the benefit of speaking after some wonderful talks, and you've already heard a lot about classical sterile insect technique. You've heard about some different approaches that are based on genetic modification. You've heard about Wolbachia technology. I'm going to continue with that theme about the Wolbachia technology, specifically in the context of what's going on here in the United States. Now, all three of these technologies, as you've already heard, are all based on the fact that male mosquitoes are very good at seeking out and finding female mosquitoes, and also the very important fact that male mosquitoes cannot bite. They don't have the mouth parts that allow them to blood feed or transmit pathogens that cause human disease. And you've been hearing in some of the other talks, some of the problems that are facing conventional mosquito control methods. Additional examples of chemical insecticide resistance, difficulty that we have in finding and treating the mosquito breeding sites. The potential for non-target effects and environmental effects. And so that's one of the truly attractive features of these different auticidal technologies is that it's species specific. The male mosquitoes that we release, they're only interested in mating with the same species. They are not going to harm butterflies or bees, and there's no known resistance. So again, I'm going to be focusing on Wolbachia. And as you've heard from previous speakers, this is a naturally occurring bacterium. It can be found broadly in over half of all insect species, as highlighted in this teaching journal. And Wolbachia is transmitted from mothers to their offspring. It's an intracellular bacterium, so it's strictly maternally inherited. And here's Wolbachia being passed in this wasp embryo. And so this is normally how Wolbachia is transmitted from mothers to their offspring. So in the United States, when we are using Wolbachia as a way to cause sterility and basically as a pesticide to control insects, and specifically mosquitoes in this case, this technology is regulated by the Environmental Protection Agency, or the EPA. So you've already heard about cytoplasmic incompatibility, so I'll just very briefly remind you. Basically, CI, or cytoplasmic incompatibility, occurs when there is a different Wolbachia type in the male. It's different from his mate. So in this example, where both the male and the female have the red type Wolbachia, it's a compatible cross and everything hatches. Same if they both have the green type Wolbachia. It's only when the male has, in this example, the green infection, and the female has the red infection, that's when you see this incompatibility, the eggs fail to hatch. And so analogous to sterile insect technique, we're able to use Wolbachia to reduce egg hatch and suppress the population. And over the last decade, we've been demonstrating this technology with two species, Aedes albopictus, shown here in yellow, and Aedes aegypti, shown in red. And here it shows the locations as well as the approximate dates of field trials that have been performed, some of which are ongoing now. We're still doing work in Puerto Rico right now with Aedes aegypti. And this data has been submitted to the Environmental Protection Agency here in the United States. And the progress with albopictus has proceeded from lab trials to field cages through what's called an experimental permit. All of this work was conducted under an experimental permit. Replicated field trials in these different regions submitted to the EPA, and albopictus has achieved full registration. Aedes aegypti is a little bit behind. We started a little bit later with Aedes aegypti, and we're hopeful to receive full registration in 2021. We're awaiting a decision from the EPA now. So if you're looking back, just how does one of these trials look? Basically, initially, all of this work originated in Kentucky, where we're based, in Lexington, Kentucky. With traditional mass rearing tools, we rear up millions and millions of mosquitoes. We remove the females and then ship the males. We just ship them commercial courier overnight to the location, whether it's Miami or Fresno, California. And then they're released. Importantly, with the Wolbachia technology, a lot of people get confused about this. We're not injecting Wolbachia into these males that are being released. It's maternally inherited. So we injected the great-great-great-great-grandmother of this colony, and the Wolbachia has been passed on from generation to generation. So we don't have to inject, we just rear them up, remove the females, and release the males. And so that's what's being done here. This is some of the early work that was done in Fresno. And I hope to highlight some ways that this technology has changed over time. But these are just small disposable cardboard tubes where we're releasing the mosquitoes into the field. This work has been continuing in Fresno. Here's an example of a field trial that was done in Miami, Florida, where we had a treated and an untreated site, both about 160 acres. Similar demographics for both areas. And unfortunately, just 10 or 15 minutes, I don't have time to go into the details for each one, but I would point out that much of this work has been published. The Miami work that I just showed has been published in the Journal of Medical Entomology. The additional work in scientific reports. And then recently a publication has come out of Nature Biotechnology summarizing some of the work that was done in Fresno, California. So I'm going to focus on this Fresno work just as the last few minutes of what some of the more recent work looks like. Again, this is published in Nature Biotechnology. I encourage you to contact me if you have difficulty in getting your hands on this publication. But in short, through these ongoing releases, we demonstrated greater than 95% suppression. So here in this yellow curve, these are the untreated sites. This is an average across multiple sites with the error shown in the yellow shading. And you can see here the normal population dynamics that occur in Fresno, California, with the population increasing in May, June, July, hitting a peak and then declining in October and November as the days get shorter and the season gets drier and cooler. In contrast, in the areas where we were releasing these Wolbachia infected males, we saw very few Aedes aegypti females. This is showing the number of females on the y-axis. Lots of males here because we're releasing lots of males, but the number of Aedes aegypti female was reduced greater than 95%. It was what's estimated. And this was done in collaboration with the Consolidated Mosquito Control District. And this work was intended to test out some additional technology that's being developed. And this is through our collaborators, Verily Life Sciences. And using robotics and artificial intelligence and computer vision to assist with the mosquito rearing process as well as sex separation. So this is a robot that's grabbing bags full of larvae, pupa, and this is a robotic arm which is manipulates the larval pans, and then uses artificial intelligence along with computer vision to recognize a male versus a female mosquito to basically assure that we're not releasing female mosquitoes. And so basically, what you can see here, these males are walking down a chute, if you will. There's a little gate here that closes to singulate the male, well, the mosquito. Camera then takes pictures of the male, defines it as either a male or a female. If it's a male, it gets put into the release container. If it's a female, it's discarded. So where it stands again is we have two different regulatory applications, one submitted for Aedes albopictus, these are called the ZAP males, and then the Aedes aegypti, this is the WB1 strain shown here. Both of these are currently under review at the Emerging Technologies branch of the Environmental Protection Agency. So, so with that, unfortunately, I see that I'm out of time. I want to just highlight that a lot of this information has been published, and so you can see much more detail. This is kind of where things stand, at least here in the United States. And again, my appreciation and thanks to the organizers for the invitation. And I encourage you, please, to reach out and contact me if you have any questions or comments. I very much wish I could be there in person and participate, but again, with events as they are, just not able to. So again, thank you for your time, and goodbye.